I found none of these. They just weren't there. Even though there's big B in the population, and there's little a in the population, I never find them together. Why might that?
Is that like for horses? No, wait, that's something.
Well, it could be like the horses. So why would one haplotypus be entirely missing in the population?
Maybe they compensate for each other. So when you have big A, big B, one of them deleterious and the other one is okay.
Yeah, so what you're describing is an example of selection. So maybe there's nothing wrong with the big B allele, nothing wrong with the little a allele, except when you find them together.
Mm-hmm.
So this one is just fine, that one is just fine. So what would that be an example of, based on the list?
Oh, that would be selection on multi-gene.
Multi-locus genotypes?
Mm-hmm. Yes.
So it's not necessarily just selection is going to see a particular locus, and maybe we'll see combinations of loci. Some will be good, some won't be good. What if this population was composed of 7 individuals? Might you have another explanation for why it comes to those?
Right.
Right. So that's a possibility. It might also be that, and this is another version of it, suppose there was just very recently this big B allele. Very recently, they were all little B alleles, and the big B alleles had just appeared in the population. Might that explain why we don't find little a ever with big B?
Mm-hmm. If it just appeared, then it has a chance to just not be with a little a. It just, like, it could be mutated and only have come from the big A or on the big A haplotype. As a result, it's not going to be mixed with the little a haplotype.
Exactly. So you can't come up simultaneously in a proportion on a bunch of different chromosomes. If it was the case that big B mutated from little b, and it's really recent, and it happened to occur on a chromosome where there was a big A allele, then it will be out of equilibrium right off the bat. That's how these things start. They can't start out in equilibrium. What if, I'm not going to change all the chromosomes, the big B, the B locus is really close to the A locus. How would that influence? Actually, I'm going to answer your last question first. So what is it that brings populations back into equilibrium? Let's go back to this. What would make it possible for me to get big B associated with little a?
Recombination of during meiosis.
So if there was a case where, say, this chromosome ended up in an individual that had that chromosome, this is a good organism, there may be crossing over. Sorry, didn't knock over my worm bucket, did you? So if this chromosome ended up in the same individual as that chromosome, it would be possible to get, by crossing over, little a joined with big B. And you would expect that to happen eventually. So then back to this, what if the B locus is really close to the A locus? What would that be?
Very small amount of A and B separate.
Yeah, or I would probably phrase this slightly differently in that it would take a long time to come back into equilibrium. So the chances, it's the same idea, but the chances of getting crossing over are much smaller here than if they're widely separate. So this is an interesting observation because you can figure out how far apart these things are. People have been doing that since the early days of fruit fly genetics. So if you know how far apart these two things are, you know that it started out completely out of equilibrium, and now it's here. Let's just give it a value of 0.1. What can you figure out that's related to what this axis is? What's this ask?
Time?
Time.
Think?
That's
You didn't look fully convinced.
I'm like, I think it's time.
So over time, because of recombination,
So,
it's gonna come back towards equilibrium. It hasn't gotten there yet. Even though I know what the recombination rate is, or because I know what the recombination rate is, and I know how far towards equilibrium it's gotten, I can figure out how long has recombination been going on since this allele started. So you can figure out not only what D is, but you can also figure out when the mutation happened.
You might have already said this, but on the study guide, it asks how come it only goes to 0.25. Right? Why is it?
So we haven't talked about that yet. So the most extreme, let's remember what D is. Does that ring bells? So you can have different combinations. You can have, when you find big A, the only time you ever find big A is with big B. And the only time you ever find little a is when it's with little b. So you find say 50% that way, 50% that way. And you never find big A with little b and little a with big B. So this would be 0. So this is the most out of equilibrium you can get. So this is when we have the largest possible values, when there's an equal number of big A's and little a's, an equal number of big B's and little b's. And the only time you ever find them is either big A with big B and little a with little b, or come out the same if you have little a only with big B and big A only with little b. So they're completely out of equilibrium. If you do this calculation, this term, these ones over here, is going to equal 0, and this one will be 0.5 times 0.5. So then you get about a positive 0.25. These ones were the only ones you found, it'd be negative 0.25. And if you find them just in the proportions you would expect based on their frequencies, then it ends up being, what is it?
What's the question?
What's the question? So let's say I set this up so you could get an extreme value of 0.5. What would be the frequency I would expect to find big A and big B if the frequency of big A is 0.5 and frequency of big B is 0.5? What would I expect if things were in equilibrium?
0.25.
Yeah, so it's the frequency chance of getting big A times the chance of getting big B. Same thing for little a, little b, 2p cubed. So it would be the same thing here. I would expect 25% of the time I get little a with big B, 25% big A with little b. If you calculate the d from those numbers, what do you get?
0.
0. So it would be 0.25 times 0.25 minus 0.25 times 0.25. So that's the equilibrium. It also works if for locus A, it was 0.8 and 0.2. On locus B, it was 0.7, 0.3. Doesn't matter what these numbers are. As long as you're finding them in the frequencies you expect, just by chance, D is going to be 0. We just started with 0.5 because that's what gives you the highest possible value for C. But, that's kind of your question is not really a biological one. It's kind of how it looks it's kind of useful to think about, because what that means is that you've got things as far out of equilibrium as possible, and you can see it when you plot things out the way they do in the flow.
Okay, thank you.
So when neutral alleles are at linkage disequilibrium, that suggests passive selection on adjacent alleles? If that's,
Yeah. So let's
Why?
imagine,
tell me more.
let's
Try not to
imagine,
check.
well, there are a couple things.
Okay.
So, and that's part of the problem with this, often there are a couple of different factors working at the same time.
Yeah.
So if we find a neutral allele, and it's at high frequency, and it's at disequilibrium,
Yeah.
what do we expect to happen to disequilibrium over time? It should break down.
Yeah.
So it could be that it's very new, and if you just haven't had time for it. But if it's very new, you wouldn't really expect it to be at high frequency.
No.
So what would cause it to come to high frequency really quickly?
The one next to it would be super awesome.
Yeah. So any new allele coming in, it's going to start out at this equilibrium. It's gradually going to go into equilibrium. If you know it's neutral, it's going to take a while for that to happen, depending on how close the low side are. But if it stays at disequilibrium and gets a high frequency, something must be increasing the frequency really quickly before it goes to equilibrium. And since you know it's neutral, it can't be selection on that locus, but it might be selection on something nearby. So it's actually better probably to think about this in visual terms. Here we've got big A, there's a neutral allele that is frequency of 0.9, but you almost never find little b with little a. It's always here. So that suggests these things are out of equilibrium, or almost always here. And it's really frequent. So I'm drawing them kind of close, but just assuming they may be even a little bit farther apart. If, as you say, A is super awesome, A for awesome, a big A is awesome. If the selection favoring this allele, it's going to drag along whatever loci next to it. So they become at high frequency, even if there's nothing really advantageous about it.
It has to be neutral though, right?
Well, it doesn't have to be neutral. You could have a positive selection going on, but then you don't know which allele is being selected. So the best thing is to say, well, I know this is neutral, and then it's at high frequency. I'll bet there must be something interesting going on with that. Other loci that are near that. So it's a way to fish around and try to find things that seem to be undergoing positive selection. And actually, there's a lot of that in humans. We have this tendency to think, oh, we stopped evolving because we've got all of our technology and so on. But humans actually have some things in particular, pretty rapid evolution. And you can find that by finding these neutral alleles, or this neutral loci, way out of equilibrium, and you can kind of find a lactose tolerance. Have you guys been talking about lactose tolerance somewhere?
No.
Oh, just
Looks
dig.
like.
So does anyone have any other questions? Okay. What is the nearly neutral theory, and why did neutralists modify their neutral theory to include some selection? I think I got this, but it was confusing at first.
Okay.
So.
Yeah, it is confusing because it's one of those things where, God, there's all kinds of stuff going on. There's generation time, there's population size.
Yeah.
So, what the neutralists said, so they did all their math, and they said that the rate of substitution, I didn't emphasize this so much in class, but maybe you remember it from the book. What, according to the neutralists, should the rate of substitution depend on?
Rate of mutation.
Yeah, just the rate of mutation.
Mhm.
Boy, is that disappointing to people who are not mathematicians who are going to think more about biology.
Mhm.
So, okay, the rate of substitution should depend on mutation rates. What does mutation rate depend on? Well, actually, let's not go there yet. Let's say,
Generation time.
what determines what drives the opportunities for mutation to happen?
New generation.
New generations.
Generations. So if you have a population that has a really short generation time, you're going to have lots of opportunities for mutations. Let's see what this would look like. If we were mapping this as number of generations, what the neutralists would say is that long or short should have essentially the same curve, because you have the same opportunity for mutations per generation. So what that would mean in absolute time, that is not numbers of generations, but let's just say years, is that organisms that have long generation times should accumulate substitutions more slowly because they have fewer generations in, say, 10 years. So you'd expect long generation time species to look like that, and short generation times to accumulate more substitutions per year. But when they looked at it, it actually looked, more or less like this. That is, whether you have a long or short generation time, you seem to accumulate about the same number of substitutions per year, or per absolute amount of time. So it seems like something's not quite working. So this is generation time. What tends to be true about organisms that have really long generation times that may be relevant here?
Small populations.
Small population size relative to short generation time, collectively, large population size. Which one is drift going to be more effective in? That big, small population size. It's got a big effect of drift. In this one, drift is not very important relative to selection. So here the frequencies are going to bounce around just based on random chance a lot more. Selection is minor. Here, it's not going to bounce around due to chance. The changes you see are more likely to be due to selection. So what Ota came up with was this idea that in all the things for a given selection coefficient, given amount of selection happening, if the population is small, then drift is important. If the population is large, then selection can be important. Is selection normally positive or negative?
Negative.
Negative. So this gets back to your first question you worried about. Why is it that you have most mutations being deleterious? So selection is going to slow down the rate of substitution, but it's not going to slow down the rate of substitution in small populations because it's drift that matters. That's cool. So large populations have short generation times, so that should make them accumulate substitutions more quickly, but they're more likely to have selection acting on them, so that slows them down. Conversely, if these are small populations, drift is going to be important. selection, not so much. And the idea is that these can more or less balance each other out, so that the rates of substitution are about the same. So, more mutations per year, more removal by selection. Fewer mutations per year, but no removal by selection. And we have the equation for a given selection coefficient, so just holding the amount of selection constant. That's the rest of this.
1 over n,
2 times n.
oh, 2n.
Oh, it's less than or equal to 1 over 2 times n. Got you.
So this is just a way of saying is that at population sizes, you'll be neutral.
To neutralize.
If, will be effectively neutral. If the selection coefficient is very small, that is, this number is bigger than that, or for holding the selection coefficient constant, the population size is very small. So for smaller the population size, the bigger this size gets, so the more chance it has to be larger than that. At which point it's going to be involved in neutral. If the population is really large, this number gets really small. So for a given amount of selection, it's going to be more likely to behave neutral. Or it's going to be less likely to be bigger than this, it'll be less likely to be neutral. Yeah. Is that command? Okay.
If it's neutral, then you have that effectively neutral.
So it will be neutral if this value is larger than the selection coefficient.
Selection coefficient.
Yeah, selection coefficient. So this is just a way of saying what is, how strong our selection. So you could have a value, let's say this was, maybe it's less abstract that way. And we've looked at selection coefficients in other contexts, but you could have numbers, high selection coefficient. So 0.04. So figure out what population size would it have to be for this to be effectively neutral? So yeah, this is not neutral because selection is going to affect it, but only at large population sizes. So that's only when this number gets small enough that this value is greater than 0.05. So there's kind of a conceptual way of thinking about it, which I think most people get. And that is that drift is important in small populations to the point that it might overwhelm selection. Selection has a much greater effect in large populations because drift doesn't have it. And then there's the quantitative part of it, meaning that you can actually calculate it at what point it's really drift that matters versus selection. So that's why it's near neutral in some circumstances, small populations, not neutral in large populations. And she came from a lab, so she came from Kamura's lab, who founded the neutral theory. And, you know, she was kind of in between camps at that point, because everybody thought selection. Then the mathematicians got in there and said, well, we can explain all this stuff, just it's neutral at the molecular level. And then she was kind of bridging the two. Yeah.
Can I ask a
Sure.
So, it's probably silly, but in terms of sex, that chapter, what is obligate and
Facultative. So first, the reason I brought that up, we are kind of hung up on sex as being so important, you know, it's a major component of life. And so it may seem to us that it's really hard to just become asexual or bisexual, but it's actually not very hard to evolve asexuality from being sexual. And facultative versus obligate is just an example of, in fact, many animals are able to do both. And they choose the circumstances under which they're sexually reproducing versus asexually reproducing. We are surrounded by facultatively sexual organisms. Dandelions, most of their reproduction, despite all the flowers we see, is asexual reproduction, but they cannot reproduce sexually. So there's nothing fundamentally important about being sexual versus asexual that you can't change. At least, evolutionarily. Oh. Did it just stop?
I've been recording it, so it's fine.
What's that?
I've been recording it, so it's
Oh,
fine.
sweet. Okay.
Sorry to the rescue.
Yeah.
All right, thank you for letting me know, Chloe. I'll just go ahead and start again. All right. So am I the only one on campus that does this? Have you guys seen this in other classes with the output picked up?
I don't
Leonard tried
No one
to-
else is using it,
There's
yeah.
like a weird,
She tried to do it through the computer, and then it didn't really work, so she used her laptop.
Okay. It's hard to troubleshoot because it starts out just fine, so
And
it's
then
not
it
just
just-
when it's starting to.
And I can give you the
Okay,
to put it.
have you heard about those lizards who were, reproduced sexually and then a bunch of females got stuck on an island and they were able to be asexual? Is that weird? Do I just make
So,
that up?
yeah, okay, so the Nematophorus, we're gonna talk, I don't know if you remember the genus. Yeah, so they're a good example of asexuality. So they're the ones, I think I mentioned this in class, that show copulations. So the females mate with each other,
Yeah,
but
But-
there's no exchange of information, genetic information.
The females mate with each other?
Yeah, they go through all the copulatory behavior because that's what starts ovulation. But they don't actually.
I heard that turtles can store sperm for like 3 years.
they can see,
Bees?
yeah, queens.
Yeah, so really long time. Yeah, lots of organisms store sperm for a long time too. There was a Komodo dragon that produced parthenogenetically, that is, produced asexually, even though they're normally sexual species. And one of the things they had to rule out was had it mated like 3 or 4 years previous and just stored sperm. But yeah, that's lots of organisms do that. That would make the whole fraternity thing really complicated in humans.
So going from the sex, and just so I'm understanding Bueller's ratchet correctly, it's so if you're asexual, as you progress on and you keep making and you keep reproducing asexually, you're gonna accumulate mutations to the point where it's like it gets to a point where it's like it doesn't work anymore. Is that what Mueller's ratchet is? It's a gradual just accumulation of mutations that because you're not getting rid of them through recombination that it messes up, or is there another?
Yeah, that's the idea, that you have, since most mutations are deleterious, anytime you get a deleterious mutation, since you can't get rid of it, it happens along with a good mutation, or a good allele of some of the locus, you can't separate them out, so then you end up dragging down good alleles, and eventually by drift, you're gonna lose, even if it's just drift, you're more likely to lose the good alleles than the many bad mutations that you're also accumulating. But if you can swap them around, selection can get rid of the bad alleles at one locus and keep the good alleles that were linked to it before. And I think your book talks about, there are limitations to how effective that is as an explanation perspective in small populations.
If that's the case, then why haven't asexual, things that reproduce asexually, gone away?
So that's an excellent question. I have a picture wrote it first, and do you remember the text that went along with it? An evolutionary scandal. There have been lots of cases of asexual, evolution of asexual lineages. They tend not to last as long. So there's the observation that they go extinct fairly quickly. And we do have the phenomenon of sexual organisms. So there are times when it's advantageous to produce asexual, if you're in stable environments, for example. But there aren't very many obligately asexual organisms. There are lots that are facultative, like Daphne, we talked about facultative asexuality. So they reproduce sexually at some times of the year, so they get the genetic diversity they need and their information, and then they reproduce asexually and crank up populations. But asexuality is not the norm, or Nematophorus probably doesn't have, evolutionarily speaking, a long lifespan. And just related to this, plants have full capacity, in theory, could have full capacity for self-fertilization. And one of Darwin's books was the various contrivances by which orchids prevent, I can't remember the rest of it, something like self-fertilization. So even in plants, they go to great lengths to avoid being sexual, or at least retaining capacity for sexual reproduction.
So genetic load, what is that?
Just accumulation of
Oh,
various alleles.
so that's
No, it's right.
Mueller's ratchet. Okay, so it's kind of connected.
Is that just how you measure it?
Is that how you measure it, or is it just sort of like it is the accumulation of that is genetic load?
Yeah.
Okay.
Oh.
You don't have any way to clean out the debris of deleterious alleles.
I can't remember what the oxpecker analogy was used for.
So the idea there was that it's easy to imagine that something's very useful for a specific purpose and think that there must be adaptations there. And we were taking the ox view of things. And the idea has been for a really long time that being reasonably tolerant of oxpeckers is adaptive because then they'll take parasites off of you. But the outcome of that was that in fact it's a mixed blessing because it would cause wounds and cause wounds to keep healing. So, you know, that argument's at best complicated. And, you know, the comparison is with the Just As stories in Days of Old, where you come up with a story and you don't really think carefully about how to test it. It just seems kind of obvious. Or maybe you are a major player in the field and your word carries weight. So suddenly that becomes the law. Now people test these things more carefully.
So can we go over QTL mapping? Not the whole thing, just like not like super quickly, just like an yeah.
So there's a limited amount that you need to know. So, for example, I mentioned lot scores. I also mentioned that the whole thing is very counterintuitive. So we'll try to go over the most important things. So what's the goal of QTL?
Isn't it define locus or gene? Yeah.
Find the locus or loci that are related to phenotypic variation. So sometimes there'll be lots of genes involved, sometimes there'll be a single gene. And one way to do that is to get individuals that are close enough that they'll interbreed with each other, but for which you can distinguish their chromosomes in some way. So the way people often distinguish it is maybe there's something distinctive about, I'll just go ahead and use the example in the book. So, lewisii might not have the same color. And what the other one is cardinalis, I think. So there are a variety of ways that you can distinguish different parts of the chromosome on each of those. So those are markers. So I've got markers. So here's marker 1. I know that there's only one kind because these things have been breeding together for a long time. So there's no variation in alleles of all of this. And Lewis, I also has marker 1. But it's distinguished somehow. Maybe the sequence is a little bit different. Oops. Let me make sure that
The same marker, I'm sorry.
it's the same marker, it's the same location. There are different ways to talk about this. I'm trying to think if I'm doing it differently from how your book does. So we're going to distinguish them by color.
Okay.
Or actually, no, now I remember what your book doing. We will distinguish them in addition to color. I'm going to do the book one better and use color plus what they have is MC and ML. So the one from cardinalis is going to be C, one from lewisii is going to be L. It's marker 1, so we'll do C1 and L. So it's the same marker, position 1. And I can distinguish between ones that came from C, so whatever cardinalis, whatever that species is, from the ones that came from lewisii. Looking good? So one marker, one locus, 2 different origins, and I can recognize which original species that part of the chromosome came from.
One on that side is the same, sorry.
On this one?
Yeah.
Yeah, so this is marker 1, and that's from cardinalis. I just wrote through both chromosomes here to emphasize that these things have been true breeding for a long time, so we know there's not a variety of different alleles here, or a variety of different alleles here. So let's give them a phenotype. And I can't remember what your book had. What's that?
There's colors.
Color? Oh, okay. All right, so these ones are blue. That works. Maybe I didn't do the book one better. So if I breed these with each other, I will get an individual that is heterozygous. Actually, let me add one more. Same reasoning. MC2, MC3. Same markers that we found over here. ML2, ML3. The markers may be neutral. They may not even give any interest to me, other than that I can recognize them as being from a particular source and on a particular region of the chromosome. So if I make these ones together, I'll get an individual that's heterozygous, and we'll have MC1, MC2, MC3. That makes sense? So now it's heterozygous. Now I just let them breed like crazy as much as I want. What's going to happen? Mixing, so there's going to be lots of recombination. So after lots of generations, I'm going to end up with chromosomes, I'll just do it because I'm going to orient them this way, that are going to be a mishmash of partly having come from blue guys, or gals, and partly having come from the red ones. So there's a chromosome. Here's another one from another individual. And so you're just scrambling up the chromosomes by this is going to have ML1. So it has this little chunk of chromosome here. That one has ML1. And this one has MC. And now this individual has that little part of the chromosome that came from that. And let's look at where's another marker. MC2. This one actually has ML2. This one has MC2. And that one has ML2. Make sense?
Should switch.
MC2, ML2, and MC.
I obviously can't be trusted with both varying color and label.
No test writing, dude.
It'd be funny if it was Mark.
No test writing.
All right, so does that make sense?
He's really
So now I know that this little part of the chromosome came from Lewisi. This one got it from Cardinalis. I've got it from Cardinalis, which is good. All right, so just to complete the story here, let's do 3. So now we'll have ML3. Now we'll have ML3, and that has ML3. All right, so now I know what the marker compositions are. What if this individual was blue, that individual was red, and that individual was blue? So we can look through all the markers. So, does the phenotype here seem to match these markers?
The middle ones.
Not exactly. So, yeah, that matches up, but that one doesn't. Does the phenotype here match the markers? That sounds pretty promising. How about here? Now, those ones are all coding for red, but 2 of these are blue. And you can make this a little bit more complicated. I've just drawn one chromosome here. You've got 2 chromosomes and say, well, if you've got incomplete dominance, when you've got 2 blue ones, is it blue, a blue and a red one? Is it something in between? And so I just try to simplify it a little bit. So what we're looking for is, are there those regions of the chromosome that seem to be associated with the phenotype more than others? So that's one question.
Would it be MC2?
So the suggestion here is that it's on 2, that 2 is associated with it. Is there any probability that this could just be coincidence, these line up, that it's just by chance?
Yeah.
So once again, as always, there's a null hypothesis that, oh, this might all be right. So there's some statistical likelihood that this is going to happen. So that's what log scores tell you. And you wouldn't want to look at just 3 individuals, 3 chromosomes. The likelihood of this happening just by chance is really high, so you want to do lots of readings, or have lots of individuals to look at. For however many readings you do, however many individuals, there's going to be some threshold level of likelihood that you could find whatever pattern you're seeing just by chance. So what's the standard kind of statistical threshold?
0.05.
0.05, all right. Well, let's do 0.05. There's some chance that this could have happened purely by coincidence. If the chance of that is greater than 0.05, well, what would you be inclined to do? Say, that's probably just an outcome of chance, or at least I can't reject that idea. If it's less than 5% likely that this could happen just by chance, then you might say, I can reject that random null hypothesis and say, well, there must be more than, there is more than a statistically expected association. So LOD stands for logarithm of odds. We're not going to go into how that's calculated. But if this is the threshold value, you can cruise along here and look at how associated are each of these markers. Maybe you have a bunch of markers here. Some of them, are going to be no different from chance. Some of them are more so. Some might even be less so. So all I want you to get out of LOD scores is that you can move along the chromosome and see are particular markers that you're looking at more associated with the phenotype than you would expect by chance. That means they have a high LOD score. And if that's true, what would you conclude?
That it's not random, that it's actually these areas causing that phenotype.
So it seems like kind of a simple question, but I wanted to see who you first guessed at those areas. So it's not the markers themselves, but that this region of the chromosome seems to be somehow associated with this phenotypic variation.
So the high score means non-random.
High score means non-random. So if you're below this threshold, just ignore it. If it's above that threshold, then you start poking around and saying, the genes are there. So maybe, there's a collagen gene there, or there's something related to pigmentation. And then you can say, well, how might that actually be related to phenotype? And in this case, well, there was if I looked in there, if I searched that section and found some genes that seem to be associated with pigmentation, that actually ties in with the phenotype. And I should say that it's not all about finding flower colors. People often do this to find human diseases. You know that there's this disease, you want to figure out what chromosome it's on, and this is one of the ways to do it. But I should also say that it's certainly possible to have multiple sites on the chromosome or on different chromosomes. What's that? What do we call that? Let's say we had 30 different sites that were statistically associated with phenotypic variation.
Link?
Yeah, because I wouldn't necessarily call it linkage. Yeah, multiple loci. So you've got polygenic inheritance, quantitative trait variation, or different things you can call it. Some of them are really tightly associated. So if you wanted to look for a gene having a big effect, you look for one that's really tightly associated with the phenotype. Some of them not so much. So they might have a little bit of influence, but not a huge one, as opposed to other ones which have a big influence.
So just the difference between those, I think I understand. So linkage would be different characteristics, whereas multiple loci, multiple places for the same character, same.
Yeah. So what else? And so this gives you a way of going through not only figuring out which genes are associated with phenotype, but which ones seem to be having the greatest effect. And of course, it could be more complicated than that because genes interact with each other, so it may not be just one, but maybe this one has a big effect and you also find particularly on another. That's why we have computers. Yes, sir.
I have two questions, or one question. It might have already been addressed because I'm very new. But there were two articles, one under the lectures that said required reading, and then there was a second one that may have been required or
Yeah,
supplemental.
I meant to pull that one out. That's the continuing evolution of genes. I think it talked about the ENCODE project and.
And you pre-highlighted sections in it?
Yes.
Okay.
So I didn't highlight that in class, and I'm not going to write any exam questions specifically related to it, but it's still useful to read. So I didn't make a big, maybe I should send out an email saying not to worry about it.
Yeah.
But I wasn't terribly upset that I might have caused people to read something unnecessarily because it's still useful.
Okay.
So maybe I'll recommend people look it over, but tell them I'm not going to write any questions specifically related to
Okay,
that.
and then there was the second article it said required reading, and it was like 5 pages of a light blue color. I can pull up the title, but is that one, we did talk about it or anything?
I can't remember now what the color was.
Okay.
So we had the Social Spider general article, and yes, I do remember there was an earlier one, but,
Jump DNA one?
so that's the one that she just mentioned. So that one I didn't intend for people to read, but there was another one that I didn't recall what that was called.
New Social Spiders, Crawford Prize 2015.
Oh, yeah. So,
And the
yes,
junk
that one's
DNA
fairly
one.
short. So the junk DNA one is the one that's most difficult reading, but I highlighted sections and I think it's useful, but I'm not going to name it. The other two, the Crawford Prize paper, it's just a word of the Crawford Prize. It gives some really nice context into Ota and the neutral theory and Lewinton's contributions. So I think those are both reading. I'm not going to ask you who was it, Ota or Lewinton that came to whatever. But I think it helps you understand why biology got all kerfuffle about it. It drove a huge amount of research, both of us. And Camaro, we probably should have read about Camaro somewhere too. I will have a paper for Thursday that I'll post that you're willing to read this sometime.
Oh yeah, what are birds with long tails? They exist.
I have some stuff, and, under reading 12 and skills, there's a couple of sections that talk about, H2 and R2 and graphing and stuff like that.
That's broad sense and narrow sense.
You guys go over that.
Oh, broad
Yeah, but
versus narrow.
Oh,
sense.
broad versus narrow.
That's what I want to know.
Oh, okay, so that H2 should be H squared.
This is
We haven't gone over that, but that's something we did in class with the ladybugs.
Yeah.
And let's do this. You know, a world of 3 variable equations is just so wonderful because you can almost always put 2 of them and then figure out the third. So in that particular case, R equals H squared
S.
S. What's that?
Selection strength.
Selection differential.
See, I'm not.
Do I ever square any numbers anywhere?
No.
No, I don't square this. Even though I said that in class, I tell I didn't square it, the square root thing. So I almost wish I was in there. And what's R?
The response.
The response of the population. So you can think this is what's a response in one generation. So you had some practice problems related to that, or at least one, where you had the kangaroo one. So you select for kangaroos, can't remember exactly what it was, but they can't jump this line. So what's the selection differential?
It's the difference between your like population and then what you want your new one to be, right? Oh, the survivors. So yeah, survivors first.
Or the selected individuals.
Yeah.
So you've got a population, let's just say in heights that they can jump, that has a mean value, T bar, and then you have a population of ones that you let breathe. That would have its own mean, which is T star. So you could also write this equation as H squared T star minus T bar. That just tells you what the selection differential is. So You might get some kind of problem if you look at the sample problems, where you know or can figure out heritability. How would one do that?
Oh, by that slope, you cross the mid offspring and the mid, or you look at the mid offspring, mid parent, and then you find the slope of that line, and that's
All right,
heritability.
so, you know, if you simplifying assumptions, the slope will tell you what the heritability is. We did that with ladybugs. Ooh, 2 hands simultaneously.
So we were asked to estimate what the slope would be on our activity.
Mhm.
So, but then we also did on Excel to get a more accurate, see how close we were or not. Do you find the slope by finding like the most, you drive at the approximate thing and take it from, like, how do you find it? If I were to take an exam, let's say on Tuesday, how
For
do I find
example.
my slope without having Excel?
So what you would need to do is tell me how you're calculating slope. So everybody's results would be different presumably, because everyone would make a different estimate. But you at least need to make it clear that you're doing what you're plotting, what the line is, and what's being divided by what to give you. So I don't really care what the actual value is.
So the change of y with change of x.
No.
That's what I did.
What is O star
Honestly, just
minus
choose 2
O bar over, like, that calculating heritability?
Yeah, so
Are we doing that? Are we doing that? That's in the book.
The O star.
that is in the book, and one of the reasons I talked about that is because for some people, that's a useful way to think about what heritability means, what responsibility, or what response means. So if I have parental generation, and I have T-bar and star. So this is the original population, these are the survivors. So I'm selecting for larger or whatever. I would expect the offspring to have their own mean. So this was O bar. In the previous generation, there would be a response after you've done selection. So look at the offspring of these.
Okay.
I can't remember. Did we use O star?
Yeah, and right, and those T's are actually P's in this
Yeah.
case.
All right.
No, thanks.
Yeah, so same idea. So you do selection on the parents, you see how much does the next generation of offspring change. If there's very little change, what does that suggest about heritability? After all, I picked just the tallest parent. Yeah, probably not much heritability. The population didn't respond. If on the other hand, when I change the parental population, if I get exactly the same change in mean and the offspring population, what would the slope of that line be? So that equals that. What's the
One.
slope? 1.
Yeah.
What does that suggest about heritability?
Maybe high.
So you can do the slope of this line. That's the way you calculate heritability.
Yeah.
But I think the simplest way. So this is a nice, convenient way to relate R equals H squared S to the idea of heritability. H squared is equal to R over S. Response on the y-axis, and S is the selection coefficient on the x-axis. So that's kind of cool. Or you can do it by the breeding experiments.
So for, let's say you give us, or we figure out the variability in the selection, using the T-bar, T-star portion. If I'm calculating the response, that would be in the next generation, right? Or do I need to manipulate it with the O bar, O star?
You don't need to do that, because that's really what you're trying to figure out. So that, you'd be talking about what the next generation is. And kind of the underlying assumption is the offspring in the previous generation were the same as the parental before you did selection.
So the only other way to manipulate that would be if I was given offspring, I could figure out the variability. But that wouldn't.
So I will tell you now that I'm probably not going to set up a question this way because we haven't really done sample problems. But it is useful this way of thinking about how heritability relates to response and selection. So one scenario may be you figure out heritability, You know what the T star minus T bar is, so what's the next generation mean going to be? Or you know how the population responded to a given selection differential, what's the heritability of the trait? So in that case, you're essentially doing this, you're calculating the slope here. Or it's just going to be R over S. What have we left out here? Oh, so you know heritability, you know how the population responded. What was the difference between the mean of the whole population and those that actually reproduced? So what was the selection differential? Great 3-term equation. So much in there. What's that? You don't even have to square anything. Were you here when I said that? Yeah, don't square anything.
It's not a parabola.
Square root it, that's the key.
Yeah,
Yeah.
definitely.
And the natural logs, be sure to put in lots of natural logs.
Natural.
And if you can fit an integral into the exam somewhere, of course, it'll be an instant error.
Natural
Anything
selection, natural
calculus.
law.
So one of the things I like about this class and this genetic stuff is that you can be pretty quantitative without a lot of higher math. So it turns out that algebra is actually kind of useful. You know, all that Hardy-Weinberg stuff. You know, this, we let some mathematician do all the math and we get to play with the result. All right, you okay? So let's see, our exam's Tuesday afternoon. I don't expect to be available Tuesday morning for questions. I'm on campus on Monday.
I'll make sure to send you like 20 at like
What's
2 a.m. on Tuesday.
Yeah, right. But I'll be around on Monday if you have questions. Probably best to email me because I've got appointments on Monday, Tuesday. Yeah, if you want to go over something, let me know.
When
I was
was
looking
it
at
again?
your screen. I thought it was one.
Oh, what did I say about which one is it?
I was like,
The salmon one?
On the 5th of June.
The 5th of June. Yeah, you know, I'm going to turn in grades late this year. I forgot to email around.
You don't need to graduate.
Let me look at a calendar, but it's not until well after the exam. But it's worthwhile thinking about. Some of it relates to stuff that's not going to be on the exam, like the life history, and other things like adaptation and so on, which are useful. So I have to look at my calendar. I can't. I'll pick a sensible date, but it'll be well after the exam.
Cool.